Uh, Good afternoon, everyone. Welcome again to our Department of Medicine academic lunch. Today we have two esteemed speakers, Dr. Ruchika Goel, who is a associate professor in the Department of Rheumatology in CMC, and Dr. V.S. Negi, who is the professor of Immunology in JIPMA Pondicherry. We are very lucky and blessed to have these two people speaking to us. Dr. Ruchika will speak about T cells and NK cells, whereas Dr. Negi will speak about primary immunodeficiencies. This is the fourth in our series of lectures. And for those of you who have missed the previous lectures, they're available on the Department of Medicine website, medicinecmcvellore.com, or you can go to the YouTube website of our department and just type Medicine CMC Vellore and Immunology series, and you can hear the previous three series. Once again, we are very grateful to Professor Debashish Danda. He is the professor of rheumatology and former head of our medicine department unit 2, as well as the former head of the department of rheumatology and CMC. He has taken this initiative to organize immunology for the general physician for internal medicine, but it can be listened to by medical students, postgraduates of various disciplines, as well as faculty for many disciplines. So we are extremely grateful to him for organizing this for us. So first, I'll like to invite Dr. Ruchika to talk about T cells and NK cells. So over to you, Ruchika, with much thanks to you. Thank you very much.
Thank you so much, sir. So I'll straight away go to the topic after a gap of 1.5 years of teaching. Okay. I believe everybody is able to see the screen.
Yes, ma'am.
Okay, fine. So the topic which I'm going to kind of discuss or give a preface to these two cells, T cells and NK cells. I mean, it's a very vast topic, but however, I think we would be able to just peep into the developmental aspects of these cells and some important functions of these cells overall in immunology. So first I'll discuss about T cells, and I'll be discussing about the development, because development, activation and differentiation of T cells, because that is the main thing which drives the function of T cells later during any immunological insult. Then we'll just touch upon subsets and functions, and a little bit hand in hand we'll talk about clinical relevance of each subset. So what makes T cells T cells? It's basically the T cell receptor. So it is very important to understand T cell receptor is actually, it is exclusive to T cells, and it is nothing, it is just something like an immunoglobulin molecule. So you have an alpha chain and a beta chain, which is bound to a membrane. So it has an extracytoplasmic domain as well as intracytoplasmic domain. But this T cell receptor by itself, by these alpha and beta chain, is not enough to drive any kind of intracellular signaling or T cell activation. So generally, T cell receptor exists as a T cell receptor complex. What is meant by T cell receptor complex? If you look here, you have alpha chain and you have beta chain, which is going to bind to an antigen. I'll come to it in a short while. And these alpha and beta chains are actually helped by many other accessory molecules on the cell surface. So which predominantly is CD3. CD3 has actually 4 chains. You have the 4 chains are there. And these 4 chains are actually driving the intracellular signaling for T cell receptor. Now, these chains, this CD3 is important because without CD3, the T cell receptor cannot function. And this CD3 is, that's why it is considered as a pan T cell marker. Now, along with these two immunoglobulin chains, which are predominantly alpha and beta in most of the T cell receptors, the CD3 chains, along with CD3 chains and these 2 chains, you also have some more co-stimulatory or accessory molecules on the T cell surface, receptors on the T cell surface, which makes it to function ultimately. So you have the main accessory chains are basically CD4 or CD8, depending on which cell are we talking about. Now this T cell receptor, if you look at it carefully, then what you see is the the two immunoglobulin chains actually do not have an intracytoplasmic domain. That means they cannot signal by tensors. They need the help by CD3 chains, which have an important intracellular signaling adapter proteins, which ultimately leads on to T cell signaling. I'm not going to discuss in detail about these proteins because I think it'll be too boring. So now, among all the T cells, when you look at alpha, almost 90% of the T cells will have alpha and beta as the immunoglobulin chains. But a small percentage of T cells will have other type of chain, which I'll come to in short while. Now, what is the function of, so because of this T cell receptor, this T cells, they are able to function. Now, the T cells, what is the function of T cell? It depends on the age of a person. So during infancy and early childhood, the T cells are predominantly involved in tolerance, in response to infectious agents, pathogens, etc. And also it is also, that is the time when the T cells are responsible for memory formation. But when you go towards adulthood, late adulthood, and around 40, up to 40 to 50 years of age, the main function of T cells in a normal human being is basically homeostasis, control of infection, immunoregulation, and tumor surveillance. And as you go, as you age, then these T cells, they acquire immunosenescence, and also they have a predominant role in driving inflammation. So this is general normal role of T cells during different stages of life. Now, if we come to the development of T cells, the T cells are basically, they are originating, they actually develop from a common lymphoid progenitor, which is basically, the bone marrow is the source of that progenitor. But the development of T cell and the maturation of T cells happens in thymus. This development generally happens during the fetal stage as well as during early infancy. So what happens is, when this common lymphoid progenitor, it goes to the thymic cortex, it undergoes interaction with a lot of local factors and thymic epithelial cells. Interaction of this lymphoid progenitor along with thymic epithelial cells and other local factors, this lymphoid progenitor gets committed to a T cell, to T cell lineage. Now, initially, when this lymphoid progenitor gets committed, it is negative for both CD4 as well as CD8. It just gets committed to a T cell, that's all. But as it goes through this cortex, it ultimately acquires both CD8 and CD4 to become a double positive T cell. And double positive thymocyte basically. And when this double positive T cell, it undergoes further differentiation and loses out one of its surface marker to become either a single positive CD4 cell or a single positive CD8 plus T cell. And then after that, it emigrates to the periphery. Now, along with this whole differentiation, there are some important steps which happens in thymus. Thymus is responsible for selecting out the T cells which are going to be helpful to a person later on during the life. So that means we should understand, I think it must have been covered already, a concept of selection. That means when the T cells enter, they are just, having all sorts of markers, and But during the course of time, like if 100 cells, if 100 cells enter, only one cell comes out of the thymus. So what happens to the other 99 cells? So basically this is called as the process of selection. Now, how does it occur? When a T cell, for a normal human being, we would want a T cell which is able to recognize our own antigens as self, and at the same time is able to tolerate our self antigens which are good to us, and at the same time it should be able to eliminate the pathogenic or the foreign antigens or the antigens which are not useful to the body. So that is what and that happens by MHC recognition. So I think there has been a series of this MHC class recognition and all. So when a peptide is presented to T cell by any MHC, the T cell recognizes that peptide. Now it is very important for a T cell which recognizes our own MHC to survive. So that is why in first selection, you have a positive selection. Positive selection means only the cells, the T cell receptors, which are able to recognize our own self MHC, they survive. So they survive, and then they get subjected to something called as negative selection. Now, negative selection is that if the T cell is able to recognize your own self MHC very, very strongly, that means it is going to kill the antigen presented by that self MHC, which is not good. We don't want our own cells to get killed. So that is why the T cell receptors which are recognizing our own self MHC very strongly, that gets eliminated. So you have death either by neglect, that or you have death because of overactivity of that affinity of strong affinity of T cell receptor to the self MHC which is presenting the peptide. So that is why the T cells which recognize our own MHC survive in positive selection, and then later on when they are not if they are able to recognize the self MHC very, very strongly, then they get eliminated. The T cells which are able to recognize MHC very, very weakly, those receptors are also of no use. So they also get eliminated. So the ones which have an optimal affinity for the T cell receptors, which has an optimal affinity for self MHC, actually comes out as the best receptor, and that is why it is the one which actually matures into a naive T cell. And this naive T cell actually emigrates to the periphery. Now, along with all these things, what happens, the T cell receptors, they gain diversity during this whole process so that they are able to recognize many, many, many antigens later on during the life. So this whole, the maturation of T cell receptors happens by two processes, which is called as the VTJ rearrangement. It happens in most of the immunoglobulin molecules as well. And also there's something called as allelic exclusion. I don't think we'll discuss about it. But as of now, we should know that the naive T cell which comes out is able to recognize self MHCs with a fair good affinity. And that is the naive T cell which comes out. The whole process for one T cell takes around 12 to 14 days. And it is the main thing happens during the fetal life and early infancy. Now, when this naive T cell, when it comes out, it will be having a T cell receptor, which is a mature T cell receptor. Now, this T cell receptor can have, as you remember, they had two immunoglobulin chains. So you can have either alpha beta chain, which is present in most of the T cells. 90 to 99% of T cells will be alpha beta positive T cells. Or a small fraction of T cells may not have that particular chain, and they may be having gamma delta T cells. Now, just to know about gamma delta T cells, because we won't discuss about them in future during the later slides. So these are the cells which are basically, they act like innate immune cells, whereas alpha beta T cells are actually the cells of adaptive immunity, classical cells of adaptive immunity. So gamma delta T cells are basically they do not have MHC restriction, and they don't recognize the peptides presented by MHC. Instead, they recognize phospholipid antigens. So they are very important cells where you have the, at the barrier sites and at the mucosal sites also, these cells are important. All right, so after the T cell has egressed from thymus, it is a mature T cell. It is a mature T cell, but it is still a naive T cell, and it is not able, it will not be doing its function of interacting with antigens and producing a immune response. So it actually has to encounter an antigen, get activated in the periphery to be able to act as an active T cell. So now we'll discuss about the peripheral migration. So what happens? The naive T cells, they continuously recirculate between the blood and the lymph node. And they reside in the secondary lymphoid tissue such as lymph node. If the naive T cell does not encounter antigen in the lymph node, it goes and recirculates to the blood. And this whole thing happens in 12 to 24 hours again. So in the secondary lymphoid organs, what happens is you have T cell activation. Now what drives the T cell activation is the antigen, which gets engaged with TCR after being presented by suitable MHC. And along with that, you also require co-stimulatory signals, which are able to cause the accessory T cell receptor complex chains also to get activated, which ultimately results in signaling. Now, the accessory chains which are important are either CD4 or CD8, depending on which cell are we talking about. And along with that, we also require some more chains, and the most important is CD28, which is actually an important receptor which drives the activation of T cells. And this particular cell is important because it is a marker. It is a marker of autoimmunity in many diseases, in autoimmune diseases, especially granulomatosis with polyangiitis, rheumatoid arthritis, cytomegalovirus, etc. So what happens actually when a T cell encounters the antigen? So this is an antigen presenting cell in its membrane, and this is the T cell. So what happened when the antigen is presented, this antigen, when it is cleaved into various peptides, it gets presented by the MHC to the T cell receptor. This is the T cell main alpha and beta chain. Along with that, you have the CD3 receptors, which is also activated. And this results in further downstream signaling. And this signaling is not enough by itself. We require help from other adapter proteins like CD4, and you have other receptors like CD45 receptor, CD28. All of them, they combine with their cognate ligands present on the antigen presenting cell, which ultimately leads to a strong signal, and that leads to differentiation of T cell into active T cells of different variety. Now, this MHC about the peptides which are presented to T cell, we will come to it. We have either cytotoxic T cells or we have helper T cells. So when the peptide is presented by class 2 MHC, it basically activates and differentiates the T cells into T helper cells. And when the peptide is presented by self-MHC, basically it is recognized by the CD8 T cells. Now, this is about the activation of T cells. Now, as we said, the T cell encounters an antigen and it gets activated and gets further differentiated. So, what is the fate of T cell after it encounters its antigen? So either it gets differentiated into effector cells. These can be either T helper cells, which are characterized by CD4 positivity, or they can be cytotoxic T cells, which are characterized by CD8 by marker, CD8 T cell marker. So when these, or it can differentiate into another cell, which is not a pro-inflammatory cell, that is the T regulatory cells. I'll not discuss about it in detail. Now, the T helper cells can further differentiate depending on what kind of signaling it receives, which we'll come discuss in the next slide, into either a T helper type 1, Th2 type 2 cell, or Th17 cells, and there are some more varieties of T helper cells which are not very well recognized, studied till now. Or it differentiates into a cytotoxic T cells. All these are basically effector T cells. These are effector T cells. Now, if the T cell is not getting converted or differentiated into effector T cell, what happens to it? So again, this is a naive T cell. Now, this naive T cell in blood or in lymph node, what happens? This naive T cell, when it encounters an antigen presented by an antigen presenting cell like dendritic cell, it gets either activated into an effector cell, as we discussed earlier in the previous slide, or it gets converted into a memory T cell. And this is basically when activated T cell gets converted into a memory T cell, it is a central memory T cell. Or what happens, this effector cell itself gets converted into, get differentiated further into effector memory T cell. So central memory T cells, effector memory T cells, or it remains as an effector cell and ultimately gets migrated to bloodstream, and via bloodstream it goes to the place of insult, like wherever infection is happening or where you have a tumor or etc., and it goes and does its job. Now, this trafficking of cell, it happens between lymph node and blood. So it will be something like you have an antigen which is presented by antigen presenting cell in any secondary lymphoid organ, predominantly in lymph node or in spleen also. And this actually activates the T cell. If it is an effector cell, what will happen? It will come to the bloodstream. And from the bloodstream, it will go to the tissue of insult and then ultimately does its effector function. So depending on whether it is a T helper 1 cell, cytotoxic cell, or Th17 cell, Th2 cell, and so on and so forth. Now, not all the naive cells actually gets converted into an effector cell or gets differentiated into a memory cell. So it all depends on the strength of the signal which it receives from antigen presenting cell which is presenting the peptide. So if it is a excessive signal or it's an insufficient signal, in both scenario, this naive T cell ultimately dies. So one is called as activation induced cell death, which is because of excessive signal, or it can die because of neglect. So it is an insufficient signal. But if it has an optimal strength, like a low to high strength, then it gets converted into an effector cell or a memory cell, depending on the situation. So the effector cells themselves can get, if they are not utilized, they can either die or they can again differentiate further into effector memory cells or terminal effector cells. Now, this is basic. These are the markers. I'll just leave the slide and you can go through it, which differentiate the T cells from central memory cells, from effector memory cells, and there's one more T cell phenotype which I'll not be showing. Now, other than as just now we saw that, so it is very important that an antigen is presented by MHC and it should produce a good signal, good enough to activate T cell. Along with the antigen presentation, we also know that you have to engage the co-receptors also by their cognate ligand for T cell to get activated. Along with that, a most important thing what happens is basically an important factor which is important for T cell activation is interleukin-2. So interleukin-2 basically is active, it acts on the interleukin-2 receptor on the T cell, and which ultimately differentiates T cell into its various phenotypes which we have discussed earlier. So ultimately, sorry, what has happened? I don't know. Sorry, just ignore the lines. So ultimately, three things happen. Three things are required. You require an antigen, you require a co-receptor engagement, and also you require the important cytokines to drive the naive T cell to become an activated T cell. Now, apart from interleukin-2, it is important there are various other cytokines which are also important, which ultimately determines whether this T cell, which phenotype this T cell is going to adopt later on in life. So along with the other factors, if you have interleukin 12 and interleukin 18, then it causes the activation of transcription factors STAT1, STAT4, TBET, etc., which leads the differentiation of naive T cell into Th1 cell. Now, Th1 cell, the signature cytokines produced by Th1 cells, which ultimately leads on to autoimmunity, are interferon gamma, TNF, and interleukin 2. If in the presence of interleukin 4 and interleukin 6, the naive T cell gets converted into differentiated into Th2 cell, which promotes allergy, and it has an antiparasitic action because of these cytokines which are released. If the naive T cell is basically subjected to interleukin 6 along with TGF beta and interleukin 23, if all these factors are present, then we have further transcription factors which gets activated, and ultimately it leads on to differentiation of Th cell into Th17 cell. And the signature cytokines which are produced by Th17 cells are interleukin 17A, interleukin 17F, interleukin 21 and 22. These are important in autoimmunity and inflammation. This cell is one of the predominant cell involved nowadays, they say, in rheumatoid arthritis and psoriatic arthritis and so on. Then there is another variety of T cell, which is T follicular helper cell. So when the naive T cell is subjected to interleukin 6 alone, there is a transcription factor BCL6 which gets expressed, and ultimately it leads on to differentiation of T cells into T follicular helper cells. Now, these T follicular helper cells, if you remember your lectures from B cells, they are important in formation of germinal centers along with B cells, and this is a cell which is important in pathogenesis of Sjogren's syndrome also. The signature cytokines which are released are interleukin 21, interferon gamma, and TNF. Now, if the naive T cell gets exposed to TGF beta, then you have expression of FOXP3, which leads on to its differentiate into Treg cell, and TGF beta, interleukin 10, and CTLA4 are the cytokines produced. And this Tregs ultimately leads on to immunosuppression, and it is just not by cytokines. There are various mechanisms which I think we can't afford to discuss at this point of time. So I think with this, I think we'll end the T cells. Yeah, before that, I just want to say what we discussed till now was classical alpha beta type of T cells. But as I've already discussed, we have a small subset of T cell, which is gamma delta T cell. It is predominantly present in skin, intestinal epithelium, and pulmonary epithelium. So these are the places, these are basically the barrier tissues where they act as more like an innate immune cells. And the gamma delta T cells, they recognize a microbial phospholipid antigen, and this is which is found on Mycobacterium tuberculosis, and it is also important in some of the antiparasitic immunity and some immunity against the intracellular bacteria. Now we will move on to natural killer cells. Now, natural killer cells, till now what we discussed were T cells, and they are the important cells in adaptive immunity. But natural killer cells in general, other than some cells which are called as natural killer T cells, these are the main cells in innate immunity. So the natural killer cells are actually, they are distinguished from lymphocytes by the presence of large cytoplasmic granules. They constitute around 2 to 18% of the peripheral blood lymphocyte pool. Now, about the development, as the T cells, the natural killer cells also are derived from a common lymphocyte progenitor. But once this common lymphocyte progenitor gets exposed to interleukin 15, which is a main cytokine driver of NK cell, this common lymphocyte progenitor gets committed to take the NK cell route, and then by virtue of, with a constant exposure to interleukin 15, these common lymphocyte progenitor ultimately gets differentiated into a NK cell. Now, the classic NK cell phenotype is cytotoxic, that you have cytotoxicity, which happens in the presence of interleukin 15, interleukin 2, and some more cytokines like interleukin 12, interleukin 18, and interleukin 21. So the cytotoxic T cell is the cytotoxic NK cell is the main phenotype of NK cell. But just for people who are interested, nowadays you have some more concepts of NK cells, like you have tolerant NK cells, you have regulatory NK cells, and so on. But we will discuss only about the classic phenotype, which is the cytotoxic NK cell. So now, basically, other than interleukin 15, which is the main cytokine, which ultimately leads on to the differentiation of the common lymphocyte progenitor into NK cell, this interleukin 15 also leads to proliferation, the differentiation. It keeps the survival of NK cells, and also it leads NK cell to gain its functional potential of cytotoxicity. Along with interleukin 15, we also require interleukin 2, which is produced by the T cells, especially the T helper cells, as well as dendritic cells. Then we also require interleukin 21, which is produced by activated T cells. We require interleukin 7, which is produced by the stromal cells or the nurse cells, and we require interleukin 4. So in the presence of interleukin 15, interleukin 2, interleukin 2, and interleukin 7, what you get is the classic cytotoxic NK cell. But in the presence of interleukin 4 and interleukin 21, basically it leads to inhibition of NK cell proliferation. But depending on which one is higher, either it leads on to increase in NK cell cytotoxicity, or it leads on to down regulation of an inhibition of the NK cell killing. So now, what drives the NK cell activation? So other than interleukin 15, which is the main cytokine, along with interleukin 2, which is involved in its differentiation, the primary drivers of NK cell activation is basically when there is a DNA damage, or it is important in tumor immunity. So when you have tumor cells or transformation of a cell into a tumor cell, or when there is an intracytoplasmic microbial infection or some stressful conditions. So when all these things happen, what happens is basically the NK cell gets activated. Now, once NK cell activates, what does it do? The classic thing is that it releases its granules, and once the granules are released, it leads to lysis of the target cell. Now, when the target cell gets lysed, there can be some of the antigens which are present on the surface or inside the target cell, they get modified, and they are presented as like neoantigens, and they basically are phagocytosed by dendritic cells and can be presented to T cells. Now, so direct cytotoxicity or an indirect effect on antigen presentation, or some of the NK cell, the other important function of NK cell is cytokine secretion. And the main cytokine, the signature cytokine for NK cell is interferon gamma. Now, the role of interferon gamma is basically the differentiation of T cells into its phenotype. Now, NK cells are basically, they're quite interesting cells in the sense they have a whole lot of receptors, and their function is totally an interplay between which receptor is activated. So the NK cells have a lot of activatory receptors. The classical examples are killer-like immunoglobulin receptors and NKP30, NKP44. So the ones which are shown in green are basically activatory receptors. At the same time, the NK cell also has inhibitory receptors, which are another type of killer-like immunoglobulins. There is a way to basically figure out whether they are activatory or inhibitory. And you have some more receptors like CD94, which is basically the receptor for HLA-E. And also there are some co-stimulatory receptors which are present in NK cell, which leads on to activation of this cell. But the signature surface markers which makes NK cell, or by which we identify NK cells in the lab, is basically the presence of CD16 or CD56 and the presence of the CD226. These NK cells are differentiated from T cells by the absence of CD3. Now, there are two types of NK cell. So, majority of NK cells are basically the cells which have very low expression of CD56. And these are the cells which are classic cytotoxic NK cells. And then you have around 6% of the cells, they are highly positive for CD56 marker. And these are the cells which produce cytokine. They are weakly cytotoxic and they do not have the classic cytolytic machinery. So what is a classic cytolytic machinery? Basically the granules, which contains main two enzymes, granzyme and perforins. So how does this CD56 dim cell kill? So it basically kills. One is just by simple degranulation. It comes, yes. So, sorry. So how does it kill? I'll just come to it in a minute. So now what happens? As I said, it has activatory receptors and it has inhibitory receptors. So when you have all these cells, all these damaged cells are continuously produced in the body. And it is not always advisable for NK cell to be activated all the time because it is a killer cell. So in normal condition, when the load of these antigens is less, what happens? The inhibitory receptors, they basically engage their ligands, which are presented by MHC class 1. So when MHC class 1 presents peptides to the NK cell receptors, the receptors which are engaged are basically inhibitory receptors. And so inhibitory receptors basically prevents the NK cell from getting activated and killing the cell. So what are these peptides going to be? Basically, they are the self peptides. So the peptides which are presented by self MHC class 1, they lead on to inhibition of NK cell and thus NK cell doesn't get activated. But if you have in the situation of stress, infection, etc., what happens is you have a lot of activating receptors which gets expressed on the NK cell membrane. Classic example is NKG2D. This leads on to active ligation of this particular receptor with the ligand which is presented by MHC class 1 like receptor which is present on the target cell. And along with that, so because of this, what happens? And along with that, what happens? The MHC class 1, which is present on a normal cell, it gets underexpressed. So when there is an underexpression of MHC class 1, the inhibitory receptors are not engaged. So inhibitory receptors are not engaged and activatory receptors get engaged, and ultimately it leads on to activation of NK cells and killing. So this classically happens when the normal cell, the normal host cell gets modified to express some different types of antigens. For example, when the normal host cell gets converted to tumor cells. So this is what happens. So NK cell, how does it kill? The most important way it kills is that it produces granules and that causes cellular cytotoxicity. It can also produce cytokines and chemokines and ultimately leads on to elimination of the pathogenic cell. Or as we discussed earlier, it leads to co-stimulation of other immune cells. So the mechanism of killing, I'll just finish in 5 minutes. So the mechanisms of killing is basically the NK cell. The classic thing is that the NK cell gets actually, it ligates with the antigens which are present on the disease cells, releases its cytotoxic granules along with some interferon gamma and TNF alpha, and it kills the disease cell. Another way, this is the natural cytotoxicity. Another way is basically cytokine stimulation. That NK cell, when it gets activated, it releases various cytokines and ultimately leads on to activation of cytotoxic T cells, etc., and kills. One another way by which NK cell kills is, as I told you, CD16 is one of the markers on NK cell, a receptor, an important receptor which is present on NK cell. And when the CD16 is basically what it recognizes, it binds to the immunoglobulins. So by virtue of binding to immunoglobulins, it basically is, it binds to immunoglobulins which have coated the disease cells. So any antibody coated cell, basically, which has immunoglobulin molecule on it, it binds to CD16 receptors, or otherwise it is also known as FC receptor. It causes activation of NK cell. It releases cytotoxic granules, interferon gamma, TNF alpha, and ultimately kills the antibody coated cell. So this is antibody dependent cellular cytotoxicity. Now, as we had seen in T cells, here NK cell also actually, whether it gets inhibited or it gets activated, it basically depends on the type of antigen which is present, which it basically ligates to. So if the NK cell receptor ligates to any antigen which is presented by self-MHC, then what happens? The inhibitory receptors are activated and you have inhibition of NK cell killing. If there is a missing cell, means a target cell is not having self-MHC or class 1 MHC, then what happens? The inhibitory receptors are not engaged and you have activation of NK cell. If you have an induced cell, means you have modified antigens which are able to ligate or bind to the activatory receptors in spite of having self-MHC, the NK cell will get activated and will kill. Or if you have a non-self MHC, then you are not able, there's no inhibitory receptor ligation, and ultimately only the activating receptors are activated, and it will definitely lead on to NK cell cytotoxicity. Now, other than the killing, the NK cells can also, so this is an activated NK cell, as happens in tumor and virus infected cells, when they ligate to this NK cells, it releases a whole lot of cytokines like interferon gamma, GM-CSF, and various chemokines. This leads on to activation of T cells and B cells. And this T cells and B cells ultimately will lead on to the immune response. Now, NK cell can also, by virtue of chemokines, activate the dendritic cells to produce a lot of cytokines, which leads on to further activation of NK cells, and ultimately it leads on to degranulation. So basically, the NK cells are able to kill by 3 mechanisms, as we discussed till now. Now, various soluble factors, I just wanted to show in this is that depending on which type of granules are released, which type of mediators are released, the NK cell can do the function. So if it's anti-tumor cytotoxicity, you have various cellular granules and TNF alpha and so on. I think this is just basically just to know that. Now, just a word about NK cell disorder. NK cell deficiency is generally not compatible with life. There are only 100 cases that have been described, and as is expected, if you have NK cell deficiency, there will be viral infections, the risk of malignancy will be there, and generally these children will not survive into childhood or adolescence, and will unless they are offered hematopoietic stem cell transplantation. Now, NK cell deficiency can be a functional deficiency, or it can be because of absolute decrease in the NK cell maturation. And these are the various disorders in which NK cell is found to be defective. Now, most important thing what you have to understand is that before you quantify NK cells and diagnose NK cell deficiency, you should definitely know the other factors which lead on to erroneous decrease in NK cells. So that is, you have various drugs like alemtuzumab, glucocorticoids, alcohol, salicylates, theophylline, various other genetic disorder, and transient suppression of NK cell function by acute stress or illness. Okay, this is the last slide. The NK cells in autoimmune disease. As in other cells, NK cells are also important in autoimmune disease. Why are they important? Because they lead on to pro-inflammatory cytokine production like IF and gamma. They lead on to B cell activation and class switching. By virtue of various cytokines, they lead on to dendritic cell maturation. They lead on to co-stimulation of T and B cells, monocyte activation and cytokine production. They lead on to cytotoxicity of immature dendritic cells. They also lead on to fibroblast pro-inflammatory cytokine production. So it is important in rheumatoid arthritis, scleroderma, etc. And they also lead on to osteoclast differentiation and bone destruction, as again it happens in rheumatoid arthritis. As of now, NK cells have been implicated best in rheumatoid arthritis, not in and in psoriatic arthritis, where these killer-like immunoglobulins are very important. In other diseases, they are still being studied. I think I'll stop with this. Thank you.
Hi Ruchika, welcome back.
Hello, sir.
Nice as you usually do. It was an excellent lucid presentation.
Thank you,
I'm
sir.
sure the trainees have benefited. Is Dr. Tambu here? Okay, is Professor Negi is around? Professor Negi, can you please?
Yeah, I'm here.
Okay, welcome, Professor Negi. We will take the questions at the end, Ruchika. So I will see the questions in the chat box and I will ask you at the end. So I ask the trainees to keep the questions typing on the chat box and I will pass it on to the respective speakers. So Ruchika is back from her 1 year stint at the Birmingham, and Ruchika has always been interested in immunology, and she has, as you have seen, has explained T cells and NK cells so beautifully. I have now our final speaker of our session, of our series of sessions rather. We started with Dr. Sita Nayak, Dr. Ramnath Mishra, Abro. So now today the last speaker is a very eminent speaker, and those who do not know him, he is Professor Veer Singh Negi. He is the head of clinical immunology and rheumatology at JIPMER, Pondicherry. Professor Negi did his DM in contemporary time, just 1 year after me, from the same institute. Dr. Negi is a very hard-working, sincere, uh, translational researcher, and able clinician. Professor Negi is recipient of IRA, Indian Rheumatology Association Oration, and you will be all happy to know that he was the recipient of the last year's BC Roy Award for eminent medical teacher. So this is he is the first rheumatologist or immunologist, clinical immunologist to get this award. So Dr. Negi has made our tribe very proud. So thank you, Dr. Negi, for accepting our. You are busy with your COVID responsibilities, and I'm very glad that you could make it. So without wasting any further time, Dr. Negi will be speaking to us on primary immune deficiencies. It's very important uh, for the trainees to understand why you chose a rare group of disorders. The reason being, the primary immune deficiencies are the nature's animal models. The nature, Ruchika has explained T cells, NK cells. Previously, you heard about B cells and all the immune arms, complement system, phagocytes. So the immune system can go wrong in T cell or B cell or complement or phagocyte or multiple components, and so many other arms of the immune system which are less described. So it is wonderful to understand the immunology. The best way is from primary immune deficiency disorders. In another word, I can say the primary immune deficiencies are the true immunology teachers for the immunology students. So you will now enjoy for the next 45 minutes. Dr. Negi will give us his wonderful deliberations. He is full of information and knowledge, updated, and he is very busy in clinical, translational, and basic research. Dr. Negi, welcome.
Thank you, Dr. Danda.
Thank you.
Thank you for your kind words. As Dr. Danda has already spoken, that this is the last lecture of the series on basic immunology for internists, and it's basically it's a culmination of all the lectures and in the form of their clinical application as Dr. Yanda said, the PIDs are the nature's experiments or nature's animal models for what can go wrong with the immune system. So, coming straight to the point, what we know or what the general perception is that probably the PIDs or primary immunodeficiencies are rare disorders. They happen during childhood, early childhood. They present only with infections, and there is no treatment for them. But in this talk, we'll have a clearer idea, a little bit clearer idea that this perception, if it is still persisting, it may not be true. So now I'll share my screen. Okay, so as has been discussed over a period of last 4 weeks, I just have a brief overview of the immune system, and one second. Yeah, so the immune system consists of various organs, cells, molecules, as well as various pathways of molecules. And the organs are primary, secondary, and tertiary. Then we have cells, as Dr. Ruchika is today talked about T cells and NK cells. So there are many cells which are part of immune system. Then there are various molecules and various pathways which collaborate and cooperate to protect against infectious organisms, as well as intrinsic threats like cancers. And if something goes wrong, some different problems may also arise. So as we know that there is a system of immunity called as innate immunity. Then we have adaptive immunity, and in innate immune system we have epithelial barriers, the mucosal barriers, the cilia, the mucous membranes, mucus secretion, and phagocytes, dendritic cells, complements, and NK cells, and various other cells. In adaptive immune system, we have T cells, which are predominant in the peripheral blood. We have B cells and the various interactions between T and B cells, which give rise to effector T cell responses or effector B cell responses to contain the various threats in the form of infection or something else in the body. So it's just yeah. So when the infectious organism enters the body or tries to enter the body, there are physical barriers, and then there is the innate immune system which tries to stop the microorganism from entering. But if it doesn't happen, then the adaptive immune system takes over, and in the lymph nodes, the further processing happens, and then the effector response happens. But if some part of the immune system is missing from the beginning, or as a part of some genetic defects, or doesn't work properly because of certain other factors, then the person has immunodeficiency. It could be primary or secondary. Secondary could be in the form of some infections or some therapy, or due to dietary malnutrition. So it does result in an unusual increase in susceptibility to infections and a predisposition to autoimmune diseases and malignancies. So this is the 2019 International Union of Immunology Societies phenotypic classification. This is not the genotypic classification of primary immunodeficiencies. There are 10 groups, and there are around 350 primary immunodeficiencies which have been characterized, and there are every day newer immunodeficiencies are being found out, and we are nowhere near finding all of them. It's going to take a long time. So it is said that the more the things change, the more they remain the same. So previously we used to have the defects of the immune system in the form of innate, acquired immune dysregulation. Although these groups are more, but now essentially the things remain the same. So there are immunodeficiencies affecting cellular and humoral. Then again, see combined immunodeficiencies, the syndromic feature. Basically, the combined immunodeficiencies have been divided into two. then antibody deficiencies, which are the most common deficiencies, then disorders of immune dysregulations. These are recent ones, and congenital defects of phagocytosis, defects in intrinsic and innate immunity. Then now the newer things which have been added are autoinflammatory disorders and phenocopies. These are what we can say now is that rather than primary immunodeficiencies, the name should be the primary defects of immune system, which can present with either immunodeficiency infections or can present with autoinflammation, can present with autoimmunity, and even allergy, atopy, and cancers. So in this, there is one terminology, phenocopies of PID, which is good syndrome. Phenocopies, you are aware that there is something called as genotype, which is the genetic constitution, and then there is something called as phenotype, which is when the genotype interacts with the environment to give rise to certain characteristic features. It is called as phenotype. Phenocopies are presence of certain characteristics which are not the part of genotype. So the manifestations may be because of no underlying genotypic defect present, but the clinical picture may appear the same. The classical example is probably being from medicine background, you people will recognize that there is Brugada syndrome, which is probably sodium ion channel defect. So which has susceptibility to ventricular tachycardia. So various factors apart from genetic factors can also lead to these manifestations. So that is what we can call that as a phenocopy. Coming to the spectrum of primary immunodeficiency diseases, as I mentioned earlier, the predominantly antibody deficiency are the predominant ones. And then we have T cells and B cells and phagocytes and other defects of the immune system, which are at a lesser frequency. So the presentation of the primary immunodeficiencies, they present with what you say as most commonly as infections. That is what we know of. But they can present with lymphoproliferation, malignancy, granulomatosis, and autoimmunity. In addition to that, some of these may be may have certain other manifestations like dysmorphic features, dysmorphic manifestations, or certain unique physical findings like absent tonsils and X-linked agammaglobulinemia or hypermobility in patients who have hyper-IgE syndrome. So these are as I had mentioned, that the scope of PID is nowadays expanding, and we have newer disorders like hemophagocytic lymphohistiocytosis, probably because of the NK, as Dr. Ruchika had mentioned earlier, the NK cell defects causing unparalleled or unchecked activation of the lymphocytes, and as a result of which there is macrophage activation, and they infiltrate the bone marrow and cause various manifestations of the HLH, which could be congenital, which could be familiar, or which could be acquired. Then we have certain disorders like Crohn's disease, which can have certain manifestations in the form of primary immunodeficiency or maybe associated with immunodeficiency, and HSV encephalitis, which has now been found to be associated with TLR3 manifestation. Then we have atypical hemolytic uremic syndrome, which is ADAMTS13 deficiency, and various autoinflammatory syndromes, periodic syndromes, which are also considered to be a part of PID, rather the primary defects of the immune system. So I have made my presentation basically most clinical, because over a period of last 3-4 weeks, you have probably learned all the basics. So how that translates into primary immunodeficiency disorder diagnosis, that will and their presentation of those diseases and their diagnosis and management, that we'll try to discuss a little bit. So this is Geoffrey Mendel classification of how the warning signs of the primary immunodeficiency. Essentially, it is the recurrent infections, severe infections, infections refractory to antibiotics, and unusual infections, which are not present in a normal person, or because of commensals organisms the infections occur. Then you should think that the person could be having primary immunodeficiency. And additional features, like there are autoimmune manifestations, there are allergic diseases, there is unexplained hepatomegaly or splenomegaly, in the form of various, combination of various manifestations, can think of primary immunodeficiency. So based on age of presentation, we usually say that the cellular immune defects, they present before 6 day early, and humoral immune defects present late because of the presence of maternally circulating IgG in the fetus, in the newborn. And when the levels go down, only then the B cell defects or the primary immunodeficiency of B cells, they manifest. Usually the T cell defects or the severe combined immunodeficiency defects present within early few weeks or few months, and then later on the antibody defects or the phagocytic defects, and further later the more than 5 years, basically the complement defects and antibody mediated defects take over. So as such, we know that IgA, selective IgA deficiency is the most common deficiency, but manifestation wise, the CVID is the one which is the most commonly diagnosed primary immunodeficiency, 44%. Then we have IgA deficiency, then we have gamma globulin due to other causes, and there is one entity called as idiopathic CD4 lymphopenia. Many patients with CVID later in their life, they develop lymphopenia also. So that terminology is called as late onset combined immunodeficit or late onset combined immunodeficiency, which is also an upcoming area. So these people have family history. Many of them are genetically, most of them are genetically determined. They could be X-linked, autosomal recessive, autosomal dominant. So these are various disorders, and single disorder could be mediated X-linked, could be autosomal recessive, autosomal dominant also, and could have a variant which is X-linked. So basic underlying thing is that there will be infections which are recurrent infections. They present atypically, usually severe or chronic, caused by unexpected or opportunistic pathogen, or infections that they fail regular treatment or which are severe, which require IV antibiotics and still fail to recover. These patients fail to recover. They have chronic infection. And another thing is those people will have unusual complications of vaccination. We have heard about BCG happening in many children because of the SCID defects. So if the person has B cell defect, then there will be mostly pneumococcal infections are happening, but any bacterial infection can happen. T cell defects, bacteria, mycobacteria, viruses, fungal infections can happen. Phagocytic defects, again, bacterial defects, fungal defect, bacterial infections, fungal infections, and viral infections can happen. Okay, so classically it has been the teaching that if the person has B cell defect, he will have bacterial infections only, whereas the T cell and phagocytic defects will have the bacterial, viral, and fungal infections. So these are some of the organisms and the immune defects which can happen. So I will not go into the details of all the things, except patients with who have Epstein virus infection and develop lympho HLH, they are likely to have familial HLH or X-linked lymphoproliferative disorder. Herpes simplex encephalitis is now being described because of TLR deficiency, and JC virus infection, PML, because of JC virus due to immunoglobulin class H recombination. So these are all the newer terminologies which are coming up in the field of primary immunodeficiencies. Okay, so the fungal infections can happen in various disorders. Similarly, parasitic infections can also happen. I'm not going to the details of all these things. Coming to the autoimmunity in primary immunodeficiency. So immunodeficiencies as such are considered to be rare diseases, but according to French National Registry, there are around 4 to 4.5 per 100,000 population. And in developing countries, this information is quite scarce. But when we come to the prevalence of autoimmune diseases in PIDs, selective IgA immunodeficiency has, if the person with selective IgA deficiency, 36% of them are likely to have autoimmune diseases. So essentially 25 to 1/4 to 1/3 of the patients with primary immunodeficiencies will have some form of autoimmunity. So the mechanism, as would have been discussed previously, is the shared genetic traits. There are certain genetic susceptibility to loci, and they have human MHC or other areas which can predispose a person. Then there is break of central tolerance, peripheral tolerance, lymphopenia, which can cause autoimmunity, and signaling problems with the signaling pathways, and defects of apoptosis and abnormal removal of cell debris, and early components of the complement defects, which can lead to autoimmunity. We classically, SLE is an example. So when should a physician consider immunodeficiency in patients with autoimmune disorders? It is difficult to discuss autoimmunity in a patient with the primary immunodeficiency because most of you will probably not encounter many PIDs. So better to discuss the PID in autoimmune disorders. So a person with autoimmune disease, when presents with severe recurrent infection, fungal and bacterial infections, or with unusual organisms, same thing what I described earlier, history of recurrent infections, long-term antibiotic use, recurrent hospitalization, failure to thrive, and a family history of PID, and poor response to conventional therapy, in addition to hematology, having hematological, rheumatological, and gastric symptoms, which are not a part of the disease, like SLE patient having certain manifestations which are not very common, that should alert you to the presence of a primary immunodeficiency disorder in a person with autoimmune disease. These diseases can be systemic autoimmune diseases, or could be organ-limited autoimmunity, or could be in the form of ATOM. So SLE, we'll take one example, like if a person has early onset SLE, more severe disease, has a close friend, family relative with SLE, and has other unusual form of rheumatological manifestation like enteropathy or lung involvement, in the background of history of recurrent skin or deep-seated infection, family history of PID, or personal family history of recurrent infection, we should think of primary immunodeficiency. These are some of the immunodeficiencies which can be seen in patients with SLE. So complement deficiency, CGD, IgA deficiency, and entity called as autoimmune lymphoproliferation syndrome. Similarly, if a child has, if somebody has polyarthritis, especially early onset polyarthritis, these people are also likely to have primary immunodeficiencies. So the ones are X-linked agammaglobulinemia, IgA, the Georgia syndrome, viscoutis syndrome, hyper IgM syndrome, chronic granulomatous disease. So various combinations of primary immunodeficiencies can be seen in these patients. The thing is early onset, and the disease which is refractory to conventional treatment should alert you to the presence of some other factor causing this problem, and PID is one of them. Similarly, the vasculitis, which tends, the vasculitis is usually skin limited, and there is absence of ANCA positivity, and blistering vasculitis is one of the characteristics. Is seen in many patients with viscoutis syndrome, complement 4 deficiency, EPSID, autoimmune lymphoproliferation syndrome, and recently described deficiency of ADA2. With in association with Dr. Rana, we had published a paper recently with PGI Nurse Chandigarh and SCPGI Lucknow. So they can present with Sjogren. The Sjogren syndrome can also have PID. Dermatomyositis can also be associated with PID. Coming to hematological, rather than the well-characterized autoimmunity, the hematological disorders are much more common. ITP, neutropenia, AIHA is seen in the diseases in CVID, hyper IgM. In fact, they may be a part of the disease spectrum of the PID. Similarly, endocrine disorders are also common. EPSID is one disorder where the patient will have hypoparathyroidism, adrenal deficiency, and type 1 diabetes, mucocutaneous candidiasis, ectodermal dysplasia. So, IPEX is another disorder okay, which is of because of the defect of Tregs or FOXP3. There is mutation in the gene called FOXP3. So similarly, the gastrointestinal features may also be manifestations of, I mean, the autoimmunity may be a manifestations of various PIDs, and ophthalmological manifestations may also be there. Cutaneous, similarly, renal is, I would like to just, as I described, the glomerulitis or glomerulonephritis or atypical hemolytic uremic syndrome, which is maybe a part of what you call as diarrhea without diarrhea, HUS happening without diarrhea could be because of ADA and TS13 deficiency. And so all that itches is not always eczema. If we take the case of eczema with eosinophilia and increased IgG, many times these patients are plain simple diagnosed to have the eczema only, because it can cause increased IgE and eosinophilia also. But again, they're going by the basic rule that if the person has recurrent infections, has atypical manifestations, has eczema which is resistant to treatment, then you should think of the person could be having primary defect in the form of primary immunodeficiency, hyper IgE syndrome, viscous syndrome, or even IPEX syndrome. So similarly, chronic mucocutaneous candidiasis also has few and warts, as Dr. Ruchika mentioned previously, can also be manifestations of primary immunodeficiency. Okay. So coming to malignancy in primary immunodeficiency diseases, I'll not go into the details. Various primary immunodeficiencies are also associated with various malignancies because of escape of the because of defective DNA repair and escape of the malignant clone of cells, and as a result of defective immunosurveillance. So coming to the how do we approach a person with primary immunodeficiency? So could be these people could be having infections, could be having autoimmunity, could be having allergic manifestations. So how do we approach them? So, but the still most common manifestation is infection, but we should not diagnose a primary immunodeficiency unless we have ruled out a secondary immunodeficiency disorder in the form of either metabolic disease or an infectious disease causing immunosuppression, or immunosuppressive agents which we commonly use in nowadays in the field of medicine, and certain drugs and toxins. They have to be ruled out, then we can go ahead with the approach to diagnosis. Basic approach is the same: history, clinical examination, and general screen for auto primary immunodeficiency. So general screen, investigative screen is very simple in these patients. We start with hemoglobin. So low hemoglobin could happen because of autoimmune hemolytic anemia, which is associated, or because of chronicity of the disease. So those autoinflammatory syndromes will have anemia of chronic disease. If a person has persistent leukocytosis, could be a manifestation of leukocyte adhesion defect, but you have to rule out the person whether he is receiving steroids or not. Then differential count neutrophilia could be because of leukocyte adhesion defect. This defect, because of absence of adhesion molecules on the surface of leukocytes, they cannot go to the site of infection and they remain in the circulation. That is why neutrophilia happens. And neutropenia, either as a result of viscous syndrome or hyper IgE syndrome, monocytopenia, eosinophilia. So these presence of these things will give you indication that person could be having this type of immunodeficiency. Coming to the platelet counts, low platelet counts are seen in patients classically in patients with viscous syndrome and B cell immune defects and the autoimmune lymphoproliferation syndrome. Viscous syndrome is X-linked recessive disorder, and it presents with eczema, thrombocytopenia, and immunodeficiency within first few months of life. classically the IgM levels are low, and peripheral smear has characteristic findings, which here it is mentioned that mean platelet volume is the lower value, and peripheral smear wise also there will be smaller microthrombocytes, what we call them. So and there may be what we call as Hall Jolly bodies, suggesting that there is TBI in these patients. So that will help to make the include or exclude the diagnosis of primary immunodeficiency. Then coming to another simple blood investigation, do simple protein, albumin, and globulin. So if there is hypergammaglobulinemia, it suggests that either person has a chronic infection or a person has chronic inflammation. Okay. So, but if there is hypogammaglobulinemia, then probably it is a primary B cell defect or a combined defect, CVID, common variable immunodeficiency, X-linked agammaglobulinemia, or severe combined immunodeficiency, or phenocopy disorder like Good syndrome. Then metabolic workup may also point towards certain things. Low calcium because of hypoparathyroidism in patients with DGORGs and upset. Hypertriglyceridemia in Shediac-Terrace and Ricci syndrome, and hyperglycemia in IPEX and upset because both of them are associated with type 1 diabetes mellitus. Now, after doing these basic investigations, we come to the immunological investigation. I'll just mention here that if in the total counts, differential counts, if you have normal lymphocyte count, so probably the immune system is all right, but you have to take it with a pinch of salt, because in various primary immunodeficiency diseases, the B cells or T cells, the numbers may be normal, but still the patient may be immunodeficient. So we advanced immunological investigations are for phagocytosis. We do what we call a dihydrorhodamine test, nitroblue test. Complement defect, we measure the complement levels, immunoglobulin levels, we measure the various IgG, IgA, IgM, and B cell function then comes, these are the levels, and then we come to the function after doing immunization. Or cellular immunity can be assessed by what we call as intradermal test, Candida test, or you can do even MON2 test. Okay, so lymphocyte subset enumeration by flow cytometry will tell you the number of cells, and lymphocyte function assays can be done using proliferative shunt assay in the form of various proliferating agents. And the final diagnosis in already known molecular defects can be found out by nowadays the easily available exome sequencing. So these are the only thing is that the immunoglobulin levels have to be measured or interpreted in the based on the age, because they keep changing. They are lesser in the earlier stages, and then later on after old age, their levels, IgG levels, they decrease. So here I'll summarize that the primary immunodeficiencies, they present with recurrent infections, they have autoimmune manifestations, they have atopy, they are not only happening during childhood, but they occur in adulthood, in adult life also, and they are not rare diseases anymore. Now more than 350 have already been identified, and new ones are being identified, and probably time has come to change their names to defects of the primary defects of the immune system. So thank you here, and if you want, I can discuss a few cases of adult primary immunodeficiency, but if the time is, I think, already over, then we can stop here also.
Please continue. It will be nice to have clinical case scenarios.
Okay,
Please
so I'll not go into, I have a number of cases, but I think I'll leave this the early onset ones. So we had this patient of a boy, age of 14 years, who had disease onset at the age of 6 months, and manifestation was chronic protected eczema, and which was not becoming all right with various local therapy. In fact, the patient had received oral steroids also. and then he had history of upper respiratory tract infections multiple times, and in fact pneumonias also, and one time it was documented to have strep pneumonia. Vaccination history, which is important in PIDs, is because vaccine can cause problems, especially the live vaccines. There was no untoward reaction to vaccination, and there was no family history of PID in the family, and he was born of a non-consanguineous marriage, and he didn't have any dysmorphic features, and he didn't have any hypermobility of the joints also. So clinical diagnosis in the form of eczema and respiratory infection, probably two were there, Biscuit-Eldridge syndrome and autosomal recessive hyper IgE syndrome. He had low platelet count, mean platelet volume was also low. Hemoglobin, although was low, but otherwise other things were non-contributory. And the immunoglobulin levels were normal. So the diagnosis was fairly simple with just moderately advanced investigation. We were able to diagnose this as a case of Biscuit-Eldridge syndrome. Yeah, so here it is to just want to bring it to the notice that the person has eczema and the recurrent infection, but it took many years for the diagnosis to be made. So as clinicians, we have to be aware of these things because of advancements in science, the things which we were not able to explain previously, we are able to explain now. We are able to find the underlying pathologies, underlying causes. So this is one of the examples where the diagnosis probably could have been made at an earlier. So management is usual. The primary immunodeficiencies, you have to take care of infections with antibiotics. When it happens, then you have to give prophylaxis. And if there are associated conditions like eczema, you have to treat the eczema. Those people who have low immunoglobulin levels, they can be given immunoglobulin replacement. Whereas in these patients, if they have life-threatening thrombocytopenia, IVIG is the drug of choice. Otherwise, the treatment of choice is allogenic stem cell, hematopoietic stem cell transplantation. I'll not go into the details. Another one is a male of 23 years. The disease onset was at 2 years. Again, recurrent sinopulmonary infections, pneumonias, and recurrent loose stools. So due to various organisms and used to respond to antibiotics. And as usual, there was birth history was normal, but he had younger brother who had recurrent respiratory tract infections, undiagnosed, and both of them did not have any untoward reaction to vaccinations. This person also did not have any dysmorphic features, no autoimmunity, allergy, or lymphoproliferative disorders. So the diagnosis, again, the hypogammaglobulinemia or agammaglobulinemia or CVID or some complement deficiency, early components of the complement. So on doing total protein albumin globulin, we found that he had low globulins. Hemogram was not contributory. So the differential at this particular stage remained again. Complement deficiency was ruled out because he had low globulins, XLA or CVID. He had all the three or four G, M, A, E, all were deficient. And CD19, or probably there was not much change. They were within on the lower side of the normal. So the diagnosis was CVID, and the genetic workup because of this COVID situation could not be done in this patient. So the management is in these patients is to keep patient infection free with prophylactic antibiotics. Dose of IVIG is required based on how he responds to the treatment, which dose is preventing the infections rather than the trough levels. The dose which prevents infection, recurrent infection, is more important. Okay, so vaccination, as for all primary immunodeficiencies, live vaccines are contraindicated, whereas pneumococcal and influenza vaccines, they do respond quite well to these vaccinations. Then we have another person, in the same group, a 17-year-old boy Similarly, recurrent febrile episodes, meningitis, sepsis. So diagnosis again was CVID, hyperIgM syndrome, or agammaglobulinemia. In this, again, the globulins were low. And what we found was all the IgA were low, and whereas the CD19 was 0. So this is a characteristic of X-linked agammaglobulinemia. So that is how we differentiate between CVID and X-linked agammaglobulinemia. And exome sequencing will confirm the diagnosis. The management, this is the management plan, antibiotics and IVIG. So coming to our last case, that the phenocopy, we had presented that in a few places. We have certain people who present, who, yeah. So he's a patient of 43 years of age. His disease onset was at 41 years. Recurrent pulmonary infections, pulmonary infections, and diarrhea. And he had osteomyelitis with one funny organism called as Alcalicins xylosioxidans. So he was perfectly all right till 41 years of age. So his vaccination was normal. Everything was normal. He had osteomyelitis of the left joint. Clinical diagnosis we had made was CVID, or probably he had HIV infection. But there was history that he had been operated for some tumor in the chest. He had thymoma of the chest. So along with thymoma, if somebody develops immunodeficiency, and he had, his globulin levels were quite low. So that is called as good syndrome, thymoma plus hypogammaglobulinemia. So he again had very low levels of CD19, other things were normal. So thank you, that's it.
Thank you, Dr. Negi. I would like now some questions to come. I cannot see in the chat box any question. I would request the postgraduates to type some questions. No question is a stupid question. You can ask any question. And Dr. Ruchika and Dr. Negi will be happy to respond. Is Dr. Mohan and Dr. Tambu here?
Yes, sir. I am here, sir.
Okay. I can't see any queries from the students. So there seems to be nothing here. It's an opportunity for all of you to ask questions and make use of the experts here. Maybe Dr. Negi, if you can just mention to the medicine postgraduates that as you have given already some illustrative cases, uh, some of the examples of primary immune deficiencies apart from CVID and maybe Job syndrome, which can come late in life and diagnosed late, and so if it will be okay if you like to re-emphasize that point. And I will also ask Dr. Ruchika to mention after Dr. Negi replies on the various autoimmune diseases, what are the immune defects, basic immune defects, if you can enumerate briefly on RA, lupus, spondyloarthropathy, few common diseases, that will be useful.
So there is one question also, sir.
Okay. Yeah,
From
right
one
now it is dropped.
postgraduate has put up in chat box, sir.
Oh yeah, I've just seen it. Yes. This is Shakshi. Yeah. Someone who is known autoimmune, someone who has a known autoimmune disease on immunosuppression and getting recurrent infections, is there any point in evaluating for PID, or when can we chalk it out to immunosuppressants? So basically, if the patient of an autoimmune disease on immunosuppression getting recurrent infection, how can you check for PID? Dr. Negi, would you like to respond?
Yeah, as I mentioned earlier, that these people, one, the disease will be unusually severe in these patients. They do not respond so well. There will be some clinical features which will suggest that this patient is not simple SLE or not simple vasculitis. There will be various manifestations. There may be primary immunodeficiency disease history in the family. And the infectious organisms may be little different than what generally these people will encounter. So in addition to immunosuppression, of course, if the immunosuppression is quite severe, these people can develop infection. But if there are other what we call them as other red flag signs, then you should definitely rule out primary immunodeficiency, because they are not so rare in these disorders.
So that's Dr. Negi has very clearly explained, and also I would like to add to that, to the trainee who has asked this question. See, what happens is every primary immune deficiency is not necessarily very critically ill patients. Like I tell you, one of the situations in lupus which might be a primary immune deficiency, as Dr. Negi has mentioned, you know that the primary immune deficiency has either broadly T cell defect, B cell defect, phagocyte defect, complement defect, and the many others which Dr. Negi has added on, the newer one of the finer deficiencies. And the presentation can be either in the form of a autoimmunity, or as you said, almost 1/3 of patient may have autoimmunity, allergy like eczema and other allergic manifestations. You may have repeated recurrent unusual infections or resistant infections, and you can have immune malignancies. So these are the broad clues one gets the autoimmune disease. One of the things we come across sometimes in lupus patients who become completely alright, respond to all treatment, dsDNA counts normal for months and years, they are asymptomatic on medication and all the disease activities, but the C4 complement never came back, usually a child or a young age lupus, and the C4 complement never gets normalized. So these are the cases you can suspect. Whether it will help, your question is quite genuine. Whether it will make a difference to the patient, I am not sure, because if a C4 null allele or C4 deficiency causing lupus-like disease, and you have treated the manifestation that is lupus, whether finding out that will help or not, more than academic interest, I am not sure. Dr. Negi can respond if you like to say.
That's you have explained quite well. That's all right. Yes.
Okay. There's one more question here. In clinical practice, when do you initiate or order immune workup? Do you wait for years, or like number of infections? Any cutoff you mean to say? And it is expensive, right? So he's asking the tests or a battery of tests to find out an immune workup is quite expensive. So when should you initiate ordering tests? Should you wait for the repeated infections, etc., for a long time, or not?
It all depends on the clinical manifestations. If there are recurrent infections and the setting is of primary immunodeficiency, we should investigate right away, because these people they not only develop autoimmunity, they develop immune malignancies. And like people with CVID, if they are treated properly with IVIG, the chances of their developing other immune malignancies are lesser. Coming to the workup, as I had gone from the basic simple investigations to the advanced investigations, you find that the normal hemogram, normal total protein albumin can tell you so many things. So, and the peripheral smear, the absolute neutrophil count, the lymphocyte count. So, based on that, you can make probably 60-70%. You can decide whether the person requires further workup or not. And then, based on the manifestations and the investigations, then you can go ahead with the further investigation, the second line investigations, which are not expensive. Most of them are flow based or ELISA based. The exome sequencing is required probably for research purpose, not for diagnostic purposes and treatment purposes. So they are not so expensive. They are in fact nowadays become routine investigations.
Dr. Tambu would like to.
No questions, sir. Yes.
So I think we have come to the end of this, and we are very fortunate today to have Dr. Negi. Dr. Negi has a lot of collaborations with the French, like our ICM or the INSAM, the French research body. He has got lots of collaborations with them, and he has got a state-of-the-art lab, and he's a good friend of mine. So it's good that the final topic has been what do you want to see in real life? How does the immune system make a difference? Some kind of nature's models which can explain it all much easier in real life scenario. Although they are rare, but they are good teachers. Ruchika, I would like to say something. Maybe the medicine PGs would like to know that suppose in one sentence how will you describe the common rheumatic disease, autoimmune diseases? What is the basic defects, like with rheumatoid or SPA or psoriatic or lupus or that?
All right, so I'll start with, let's say, ANCA vasculitis and all. It is specifically an autoantibody mediated disease where the antibodies are pathogenic and they lead to inflammation. Whereas, and similarly, even lupus, in addition to specific antibodies which have a pathogenic role, like anti-DSTNA and so on, you have, in lupus, I think I would not say any of the cell is not important. You have antigen specific T cells, you have antibodies which are pathogenic, and at the same time the ultimate thing what you see is the classical complement getting activated and getting consumed. So that is what synopsizes lupus. But if you get an autoantibody mediated phenomenon, you would not suspect, let's say arthritis, you would not suspect it to be spondyloarthritis, because that is a totally different disease where you have, it's an MHC class 1 mediated disease, and you have a lot of Th17 cell mediated pathology which is happening in spondyloarthritis. And there's a lot, there is a part of innate immunity also comes into play in spondyloarthritis. Then rheumatoid arthritis, I'll say it's everything. All the cells are important, the B cells, the T cells. What else? So, and if you have certain immunological disease, certain vasculitis like HSP and all, where you have alternate complement pathway which is getting affected rather than the classic complement pathway. So as of now, what we deal with, all autoimmune diseases are diseases dealing with adaptive immunity, with varying contribution from innate immunity in different diseases. The highest being in some diseases like Behçet's, spondyloarthritis, where you have innate immunity also playing a role. That's all, sir. I don't know.
Thank you. So Dr. Negi also has DM program like us. He also has DM program. I think he also has about 4 per year. Dr. Negi, is it right? You have four seats per year?
2 per year.
2 per year, okay.
In January session.
Okay. So it's another good place to work, and so I cannot see any other questions. I now hand over to Dr. Tambu, and I appreciate Dr. Tambu for asking me to arrange it, and I myself have benefited a lot from this, and it was good to see Dr. Sita Nayak, Dr. Mishra, Abro, Professor Negi, Ruchika all in one platform, and I think everybody has done their best to make these difficult areas as simple as possible, and I think it will definitely sensitize uh, some of the trainees' minds, and maybe some of them will join rheumatology, take up as their career. Thank you, Tambu.
It's my very pleasant duty to thank Dr. Debashish first. So only two words, thank you. We put a challenge to you to train a teacher's immunology for a physician's level, and we have more than exceeded our expectations. So thank you very much, sir. We are very grateful to you. Thank you to all the experienced teachers you have asked to take. We can't thank each one personally today, but please extend our thanks to each one of them. Department of Medicine is very grateful for them. I know that your brother wasn't well during this time with COVID, but despite that, with a brave face and so cheerfulness, you have been with us through all 4 sessions of 8 speakers. Thank you. That's really very wonderful. So please give our regards to ma'am and your son, and we thank the entire your whole team you have put together. The Department of Medicine has been greatly blessed. So we'll share with you the YouTube link so you can share it with the other postgraduates in rheumatology wherever they require. So we are thankful to you again, sir.
Thank
So
you.
with that, I wish all of you. My duty also to thank Mohan and the telemedicine team for organizing this whole thing. Thank you, Mohan. Thank you to the telemedicine team. So with that, sir, thank you very much, and wish all the participants a very good evening. Thank you again, Ruchika and Dr. Negi, sir, for today. Thank you.